Hello friends, I am Gaurang Makwana. I welcome you all to my another video tutorial biotech guide. So in today's video lecture, we are going to discuss about a evolutionary highly conserved process which is also known as a post-transcriptional gene silencing, that is a RNA interference. In this video lecture, we are going to discuss about basically first two experiment which having a correlation with RNA interference, then basic introduction, after that two methods RNA interference. First is RNA based RNA interference and second one is a DNA based RNA interference. In RNA based interference, we are going to discuss about RNAi by siRNA, small interfering RNA, and second one is RNAi by miRNA, microRNA. So and last also will cover the application of RNA interference. So today's video going on very interesting level. Stay tuned with us and enjoying this video lecture. So let's start first. Here you have one flower, petunia flower, and you inserted pigment inheritance genes. So what are the possibilities? So basically two possibilities: less pigment or a combination. So what happens? Resulted less pigment. How? So basically researcher found mRNA is reduced in less pigment of a flower. So basically the main reason is mRNA. And another fact is plant having a RDRP gene, RNA dependent RNA polymerase, which produces a dsRNA, double stranded RNA molecule that suppresses the pigment in a petunia flower. So this is the first experiment which correlates with the RNA interference. Another experiment carried out by scientists Andrew J. Fire and Craig C. Mello. They introduced a double-stranded RNA into a worm C. elegans and resulted mutant strand of C. elegans which showing the twitching behavior. Their study indicates dsRNA is the responsible for gene silencing. So here we focusing on a dsRNA where the process whole RNA interference process is carried out by the basic or the first primary molecule, that is a dsRNA. Now let us discuss what is RNAi. So as we discussed is short for RNA interference, and it refers to a phenomena where small pieces of RNA can shut down protein translation by binding to the messenger RNAs that code for those proteins. So we know central dogma where DNA converts into the mRNA and mRNA converts into the protein by the translation process. So here in the translation process RNA interference came where small species of RNA that can interfere the process of translation and degrades the messenger RNA. Then on another hand, species terms like when the process is carried out into the plants is known as a post-translation gene silencing PTGS in a fungi quelling, and in animal is defined as a RNA interference. Now we'll discuss the methods. So basically as we discuss evolutionary highly conserved process of post-transcriptional gene silencing where dsRNA cause a sequence specific mRNA degradation. So primary molecule is what? dsRNA, double stranded RNA. The small pieces of RNA that enables RNA interference come into two varieties. So basically dsRNA further converted into two varieties of RNA, that is a small interfering RNA and the microRNA. Here microRNA, miRNA, is a short non-coding RNA nucleotide produced from the chromosome inside the nucleus and have a hairpin sequence which folding them into a double strand. On another hand, siRNA, exogenous double-stranded RNA, which is similar to miRNA but not produced inside the cell nucleus. So basically, these main difference are there between two siRNA and miRNA. Both varieties are pieces of RNA approximately 22 nucleotides long, but they differ in their role, specificity, and how they are synthesized. So basically, there are mainly two methods are there in RNAi technology: RNA-based RNAi, where two basically siRNA or miRNA. On the other hand, DNA-based RNAi. Okay, so let us discuss one by one. First, RNA-based RNAi, in which first RNA interference by small interfering RNA. Basically, four components are included: dsRNA, dicer enzyme, RISC complex, RNA induced silencing complex, and argonate 2 protein, which is a component of a RISC complex. And then three steps are there: formation of siRNA, then interaction with the RISC complex, and third degradation and cleavage of a mRNA. So first, dicer recognize and cut the dsRNA into 20 to 25 base pair long fragment is defining as a siRNA. Now this siRNA interacts with a group of enzymes known as a RISC complex, RNA induced silencing complex, with the endonuclease argonate 2 component. Now here you can observe the two strands in siRNA. One is known as a passenger and another is known as a guide strand. Okay, so here as we discussed, argonate 2 protein, which is associated with a RISC complex, which cleaves the passenger strand. In contrast, guide strand or antisense strand remain associated with a RISC complex. Okay, one strand is degraded and one is remaining strand is known as a guide strand. Now the guide strand is bind with a mRNA, which having a complementary sequences. Okay, so basically the guide strand is bind only that active site of a mRNA which is complementary to it. And then third, degradation and cleavage of mRNA, where argonate protein is cleaves the target mRNA and specific gene silencing occur. So this is the RNA by small interfering RNA, siRNA, or also defining as a siRNA mediated gene silencing. Second, in RNA based RNA interference is RNAi by microRNA, where basically all steps are same first except first formation of miRNA. So basically, as we discussed in a difference, miRNA is produced inside the nucleus. So basically in the nucleus, RNA polymerase 2 forms a pre-miRNA from the miRNA gene. Now this pre-miRNA cleaved by microprocessor complex DROSA and DCGRB and from a precursor miRNA. Okay, so basically miRNA gene to pre-miRNA and pre-miRNA to precursor miRNA. Now this precursor miRNA is subsequently transported by exportin 5 from nucleus to cytoplasm, and in cytoplasm is further proceeded by dicer in 20 to 25 nucleotides. So now process is same, interacts with a RISC complex, okay, and then degrade and the cleavage of a mRNA. Okay, so here as described, miRNA interacts with a group of enzyme RISC complex and argonate 2 protein is there, where argonate 2 cleaves the passenger strand. Okay, and guide strand is remaining. Now guide strand is doing what? Guide strand is bind with a target mRNA which is complementary to it, and then degradation and cleavages happen. Now argonate protein cleaves the target mRNA and comes specific gene silencing. So this is the process of RNAi by microRNA, or also known as a microRNA mediated RNA interference. Now role of RNA based RNA interference in a cell. So basically in regulatory mechanism, particular gene and protein synthesis by adding a layer of a control mechanism, and second one is protecting cells from exogenic parasites like viruses and from endogenic nucleic acid. So these are the role of RNA interference in a cell. Now move on in another method, second method, DNA based RNA interference. Okay, where basically In DNA-based gene silencing, a short DNA sequence is used artificially to target specific RNA molecule by activating the cell's natural RNAi pathway. So here basically in second method, what happen? DNA is further converted into the self-complementary double-stranded RNA, and that enters into the RNAi pathway. First, introduction to DNA into cell. Okay, so basically DNA is introduced via plasmid vector, viral vector, or agrobacterium mediated transformation. Then now introduced DNA is converted into further short hairpin RNA, long dsRNA, and artificial microRNA, and then after this formatted RNA further proceed into a RNAi pathway activation, where dicer is processed shRNA or dsRNA into siRNA, and that siRNA are loaded into the RNA induced silencing complex and guiding it to target mRNA for degradation or translation repression. So here you can see in the diagram here DNA which is introduced into a cell via vector, then after that DNA is converted into dsRNA. Now that dsRNA is further proceed into the another types of 20 to 25 nucleotide shRNA or siRNA or microRNA, and then now this RNA is further proceed into degradation via using RISC complex and argonate protein. Now let us discuss application of RNA interference. So various applications are there both in biomedical research and functional genomics. First, plant genetics and agriculture. So it can be employed to create plants with a specific traits, like making them resistant to pest or producing decaffeinated coffee. Then RNAi in therapeutics. So basically it is a new area of therapeutics where scientists have started to tap the potential of a gene silencing by targeting mRNA. It has shown a potential to treat disease by targeting specific genes associated with a disease such as HIV, hepatitis C, and respiratory disease. Then drug development. It is used to identify genes that could make cells resistant to certain drugs, or genes that are affected by specific drugs, providing insights into their mechanism of action. Then in cancer therapy, it holds the potential for treating cancer caused by mutated genes, which can be targeted and suppressed by using RNAi. And last, research in a functional genomics. So it helps in a quickly understanding the function of different genes in various organisms, and it might be useful in studying genes that are newly discovered and not well understood. So this is the application of RNA interference. This is all about of RNA interference. If you like my video, subscribe to my channel, press the bell icon, never miss an update. Thank you so much, friends.